So we're picking up here, again, this is Bio 212 at Norwalk Community College, and we're starting at the histology of the renal corpuscle. And again, I'm going to just begin by saying blood flows in through this afferent arteriole, travels in to the capillary bed here, but notice the capillary bed is surrounded by these things that are called podocytes. And what I want you to notice is your textbook hasn't mentioned these things, mesangial cells. Notice where you find them throughout this entire corpuscle. Whatever fluid is pushed out of the capillary bed that isn't immediately taken back up into that efferent arteriole is now going to travel through our convoluted tubules. This is the proximal one. We'll go down the descending limb of the loop of Henle, ascending limb of the loop of Henle through the distal convoluted tubule, and then into the collecting duct. Okay. Also, I wanted to point out that for whatever reason, I wasn't able to close caption part A. I'm going to try again with part B, and it just seems to me as though there's something wrong. I spent about half an hour trying to troubleshoot it, but haven't been able to get it to work. So, okay, the renal tubule and the collecting duct. So, students get lost here, and they get lost because I'm going to draw the corpuscle here. So here's the glomerular capsule, right? Wham, glomerulus. And in red, blood is going to enter in. Blood enters in. Yay. Create blood vessel. And then blood exits. Or here is our afferent. Here is our efferent. Arterioles. What we're going to be talking about is the tubule portion, though. So I'm going to continue to draw this piece here, where descend, ascend, travel around, give you some and then we're going to travel down, right? Cool. Where this is the proximal convoluted tubule here, descending limb, ascending limb, and this is the distal convoluted tubule. So let's go dist. Cool. Why am I now drawing this? I'm drawing this piece because I'm going to change colors to that sort of yellow-brown color. And where people get confused, right, what actually happens is they forget that the whole purpose here is blood moves into this space to be pushed out. So I'm going to give it a plasma color. So I'm going to sort of go like blood plasma, all right? So it's pushed into this space. There's a series of forces, like everything inside this class. You're going to follow the forces, and then it's going to travel through the tubule. And at distinct points during this trip, I'm going to try and keep it inside the tubule here. So it's like drawing in grade school. I'm going to stay inside the lines. Oh, almost. Oh, I failed. Oh, well, nothing new. Okay. And what I want to make sure you're paying attention to, aside from the fact that I actually have drawn lines for you here, right? So let's change the color here again. What we're setting up is a discussion for who gets collected back out of the filtrate, okay? And then is brought back into this space, okay? Who is taken out of the filtrate here, or what is taken out? What can be taken out here, and how is that going to happen? So, having drawn that for you, the juxtaglomerular apparatus helps regulate blood pressure inside the kidneys. So where is that? Okay, so that's back here. So it's the mixture of where you actually have the afferent and efferent arterioles, and we run literally the tubular structure right next to it, and it has this part that's actually called the macula densa. I'm just going to put a line underneath that. Okay, and the final part of the ascending loop of Henle, and inside of it you have juxtaglomerular cells. Okay, and they are going to have modified smooth muscle fibers. And inside of here, the part that should be discussed but isn't as well as you actually have the ability to modify those using nitric oxide. Okay, nitric oxide. The last part over here, we're going to put some cells in here. We're going to have principal cells. I'm going to put P cells. Okay, and I'm also going to put intercalated cells. intercalated cells. Intercalated. Intercells. Right? And what's going to happen is these are going to be the receptors. So these cells are the targets of ADH. And aldosterone. LD. Okay, so now this is where you actually have that sort of moment of clarity as to way back when inside chapter 18, when Mother Nature said, look, you need antidiuretic hormone and you need aldosterone. Well, aldosterone regulated the movement of ions, ADH regulated the movement of water, and now you can see there's a specific target. Okay, you'll see how that happens a little bit later on. Okay, but let me just make sure you get this again. Blood flow moves in, filtrate travels out. Throughout different parts of it, we're going to regulate the movement of materials in and out of that tubule. But now what I've told you is juxtaglomerular apparatus, put a big circle around this for you, okay, is going to modify smooth muscle fibers. So that means increase or decrease the flow rate into the capsule. And if you've increased or decreased the flow rate into the capsule, we also have the distal part of the convoluted tubule and the collecting duct, which has principal cells and intercalated cells that are going to have specific roles for us to pay attention to. Okay, let's get rid of that. Let's move on to the next slide. So what's actually happening here? Well, we're going to filter. We call that glomerular filtration. We reabsorb. We call that tubular reabsorption. Materials being taken out of the tubule, meaning reabsorption is being moved out of the tubule into the space where it's going to be returned to blood, right? So I'm going to put that there, put an underline underneath that for you. And this is the material that stays inside the tubule. Okay, so when we finally get rid of it, as it's exiting the body, okay, it's glomerular filtration plus secretion minus the reabsorption. So this is the piece that's gone back into the blood. Okay, what does that look like? Afferent arterial filtration occurs inside the renal corpuscle. When that happens, goes inside the renal corpuscle. And what's going to happen is the green here, that's that yellow line I was drawing earlier in my earlier cartoon. And reabsorption goes back into the blood if it's next to a peritubular capillary bed or if it's next to the vasa recta. And here's the part that can happen as well. While you're out here, anything that's inside the blood we don't need, you can take it back into the fluid inside the renal tubule. Okay, so the filtration doesn't just occur, most of the filtration occurs here. You can actually have reabsorption and secretion back into the tubule as it travels from its beginning to its end until it can no longer accept new materials. Okay? So what's actually going on inside that glomerulus? So it has about 180 liters that are pushed into those little capsules, okay, and 99% of it is returned back to the blood plasma via reabsorption. Okay, so it's going to go right back into the bloodstream. So as I said earlier, it seems kind of inefficient, but it's actually really efficient in the way that it's designed. Remember I told you actually have capillary beds. These are endothelial cells from glomerular capillaries, which means you're looking at a fancy capillary bed. And they have those podocytes. You'll see a picture of the podocytes in a second. And it prevents filtration of most plasma proteins. Remember inside lab when we were looking at the blood tests, there was one there for protein level. Okay? Plasma proteins shouldn't exit. They should be too large to actually get out. Blood cells shouldn't get out, and platelets shouldn't get out. So the fluid actually has to pass through those endothelial cells, fenestrations, and the basal lamina between the endothelium and the podocytes. You'll see how that looks in a second. And the volume of fluid is large because of the large surface area. Remember I mentioned that just a moment ago. And here's our cartoon. So here's what you've seen so far. Remember, blood flows in, passes out, and you're like, well, how the hell does this shit get out of there? Well, this is how it happens, right? Take a look at this glomerular endothelial cell. This is a fenestrated capillary bed. Fenestrated. And that means you actually have large amounts of space where material can pass through. Okay? And then you also have this basal lamina of the glomerulus, which prevents the filtration, or I should say makes sure that the filtration of material can't actually get out in a way that's going to glom things up. I don't know. Okay? And then finally you actually have the podocytes. The podocytes create these slits that are going to allow for less resistance, so whatever fluids come through, you can actually get it all the way through. So I told you a moment ago there's going to be some pressure inside here, and we'll get to the figure for that in a second, but let's just draw something quickly for you. Remember, in red, we're going to have a capillary bed, afferent, ding, 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 ding. Right, now you know it's a special capillary bed, so let's put the glomerulus around that. Glomerular capsule, let's go like this. We'll put the proximal convoluted tubule aperture opening here, right? And what's going to happen is, as fluid comes in, you have glomerular blood hydrostatic pressure pushing outward. Capsular hydrostatic pressure pushing backwards. And blood colloid osmotic pressure pulling things back in. And that's actually how you get materials right back into the efferent blood flow out. Whatever is remaining, remember, this is just an area of lower pressure now. It's going to travel into this space, and that's filtrate. Okay? Makes sense? Hope so. Okay? Now, that blood colloid osmotic pressure, it's a good chance for us to review for the final exam, where you're looking at the presence of proteins that were left behind. So whatever fluid can be taken back up will be taken back up. And this is what it looks like inside your textbook. Let's first get out of here. There you go. Again, notice here's our afferent arteriole. Again, notice here's our vasomotor nerve. You have a specific value of 55 millimeters for glomerular blood hydrostatic pressure. It's going to push out. Here's our capsular hydrostatic pressure. It has 15 millimeters of pressure. What the heck? Pushing back, and then you have the blood colloid osmotic pressure. And what's going to happen, the filtration, this is what's NFP here, net filtration pressure, is about 10 millimeters. Let's see what I can do with that. Okay. Let's do that. Let's do... Didn't want to do that. Let's go like that. Okay, well, make sure. So 55 minus 15 minus 30 gives you 10. Okay. Notice what they did here as well. They took out those mesangial cells. They took out the mesangial cells here, and they took out the ascending portion of the loop of the limb of Henle, right? Which means they also took out the juxtaglomerular apparatus in this cartoon. Okay, so I just told you how it's going to work. It's going to be pressure. How do we regulate it? That's what's most important to you, right? Because if the pressure's too high, hypertension, materials pass too quickly. They're not reabsorbed. Too low, nearly all are reabsorbed, and some waste products are not adequately excreted. Okay, so you need to regulate this. You have renal autoregulation, primary mechanism, right? Kidneys themselves maintain a constant renal flow because you actually have a myogenic mechanism that occurs when stretching triggers the contraction of smooth muscle cells. So as blood flow moves in and begins to stretch it at the afferent arterioles, it's going to reduce the glomerular filtration rate here. Okay? More blood flow moves in, stretch. Afferent arterioles will then make sure they contract. Okay? Now, what's going to happen, of course, is you also have this tubuloglomerular mechanism where the macula densa provides feedback to the glomerulus, and it inhibits the release of nitric oxide. Now, notice what's happening here. That means that those mesangial cells are making nitric oxide. The tubuloglomerular mechanism inhibits this, which then allows the afferent arterioles to constrict to decrease glomerular filtration rate. So both myogenic and tubuloglomerular mechanisms are used to reduce or decrease glomerular filtration rate. Here's the picture from your textbook that looks at the juxtaglomerular apparatus, which decreases the secretion of nitric oxide. Okay. Now you also have neural regulation. And remember, I've been sort of making sure you pay attention to that because you actually have sympathetic autonomic nervous system fibers that are in place, and they're going to release norepinephrine, causing vasoconstriction. Things can be inside your body titrated. With moderate stimulation, both afferent and efferent arterioles constrict to the same degree. Okay? So moderate stimulation. Greater stimulation constricts the afferent arterioles more, and therefore the glomerular filtration rate will drop. This is when you're getting scared, right? Mother Nature has just scared you. She constricts the afferent arterioles, glomerular filtration rate drops. This is an adaptation for some amount of decrease in glomerular filtration rate. This is the course of your day and you haven't had a chance to drink any water or go to the bathroom. Okay? A moment ago, I told you actually can regulate all of this using two hormones. Look, we spoke about previously, though, ADH and aldosterone. Notice here, angiotensin 2 and atrial natriuretic peptide angiotensin 2 reduces glomerular filtration rate because it's a vasoconstrictor for both afferent and efferent arterioles. Think RAA pathway. Atrial natriuretic peptide increases glomerular filtration rate. By stretching of the atria, it's going to allow for the capillary surface area to increase for filtration. Now, a moment ago I told you that seems like we didn't spend enough time on regulating. So there are three parts. Make sure you pay attention to that. It's renal autoregulation, neural regulation, and hormonal regulation. Okay? For glomerular filtration. The earlier hormones, ADH and aldosterone, were for reabsorption. Okay, cool. Now, so we're going to move on. We're going to try and knock out 2 minutes here, see if we can get a little bit further along than we are. Well, you're going to see that most of the materials are going to be collected. Okay, and reabsorption is going to be back into the bloodstream, both active and passive processes, and secretion of materials is going to control our pH inside of our blood because we're going to try and trap as much hydrogen, proton, or basic structures, nitrates, things like that, as best we can. How do we do it? You have paracellular reabsorption, and or you have primary active, secondary active transport of materials, usually such that something else is going to follow. So sodium-potassium pump means chloride will follow. That also means water will follow. Okay, let's see if we can demonstrate this to you. So here is paracellular reabsorption. Here is the yellow stuff I drew. This is inside the tubule. Here's your tubular cell that's making it up. And you can have sodium paracellular reabsorption traveling through here. Or you can have sodium transcellular reabsorption traveling through a cell. And notice this requires primary active transport. But the point is going to be this. Now you have sodium outside here, which can be taken up into the peritubular capillary bed. Who's going to follow? Water, we have to hope, right? Now, that's the proximal convoluted tubule. And I'm not going to read through this entire slide. There's a lot of information on here. There are a couple things you want to pay attention to. Most solute reabsorption involves sodium, which means there's going to be some symporter for glucose, amino acids, lactic acid, phosphate, sulfate, depending on what we need. Okay? So we actually take that stuff back up into the bloodstream, but we also use it as an antiporter, so hydrogen protons can be secreted out. We also want to make sure that most of the water inside the proximal convoluted tubule, okay, is actually going to be collected here. Okay? And we use a specific protein for that, and that's called the aquaporin 1 channel. And I'm not going to go through that figure there inside your textbook. We'll cover it inside class. But if you're doing this at home and you're not inside my class, notice what's happening here. Symporter, antiporter here, right? Antiporter. Sodium goes in one direction, hydrogen protons go inside the other. Look down here, our favorite friend, carbonic anhydrase. Notice you move bicarbonate into the blood as you trap water, metabolic water out here, with hydrogen protons trapped inside of it. Now the loop of Henle itself was going to travel through that. We'll pick up here in a second. This is the 20-minute point. Again, I'm going to try and see if I can get the closed captioning to work, so this might take a little bit longer, but that's where we're going to stop.